Mendels findings in modern day terms. We've already talked a little bit about how there are dominant alleles and recessive alleles. So let's go over those two definitions real quick. Dominant alleles are indicated first by writing the first letter of the trait as a capital letter. This is a trait that tends to be expressed in a person or in an organism. The recessive alleles are those that are not expressed if there is a dominant trait that is expressed. And these are indicated by writing the first letter of the dominant trait, but it's going to be lowercase. So for instance, if we're talking about red flowers and that is the dominant trait, we say that that's going to have an uppercase R, whereas if white flowers are the recessive color, that's going to have a lowercase r. It's also very important to understand that we can have alleles of a particular gene that are both homozygous and heterozygous, and depending upon what those look like will give us what is expressed and what is not expressed. So if the two alleles of a particular gene present in an individual are the same, the individual is said to be homozygous. So meaning that we have two big R's and two little r's. We have two big R's, that means it's going to be the dominant trait. Two little r's are the recessive trait. If the alleles of a particular gene present in an individual are different, the individual is then to be said to be heterozygous. So for example, if we have a big R and a little r, this is two different types of letters. So that's a heterozygous trait. One big R, one little r. In heterozygous individuals, only the dominant trait is expressed. So in this case, it would be red flowers because it is the dominant trait. The recessive allele is present but is unexpressed. So in further reproduction, this individual could possibly give a little r, so they could give that white trait, but it is being expressed right now as red. The set of alleles that an individual has is called its genotype. So your DNA codes for genes, and these are what's represented as we were just talking about, the big R, big R, big R, little r. This is your genotype. This is what your genes say. The physical appearance of that trait is called the phenotype. So in this case, with the big R, and even big R, little r, that's the red flower color. So your genotype is what your genes say, while your phenotype is what your physical appearance looks like. So I remember it as genes for genotype, physical for phenotype. Your phenotype is determined by which alleles are present. So for example, if we take a look down here at the picture, your genotype here for these pea plants, big P would represent purple flowers. This is also homozygous. Your purple flowers also could be represented by big P, little p. The little p is not expressed, that's our recessive trait, so it's hidden behind the purple one. Now if this individual is little p, then it is white because it has two recessive traits, and both of those would mean that there's no dominant to take over, so that's why the recessive is expressed in this case. So let's talk about the principle of segregation. Now this first law of heredity describes the behavior of chromosomes during meiosis. So during this time, homologous chromosomes and the chromatids are separated, if you remember back to Unit 5. Now this law, the principle of segregation, states that the two alleles for a trait will segregate or separate when gametes are formed. This is before, way before fertilization actually happens. So this happens in meiosis. So the two alleles that are together in your body cells, they will separate from each other during gamete formation, meaning that each gamete only receives one version of that gene. Now this is extremely close to the principle of independent assortment. So it's very important that you understand the two differences here. So if you take a look at this law of segregation or the principle of segregation, here are our body cells. During meiosis 1, the homologous chromosomes are going to split. During meiosis 2, the sister chromatids will split. Notice that each of these in the starting cell, we have big A, little a, little a. By the end of meiosis 2, have split into individual gametes. So now each of these gametes has only one version of that cell, or I'm sorry, of that gene. So big A here, little a here, little a here. So law of segregation happens during gamete formation in meiosis. Now the principle of independent assortment sounds very similar to the principle of segregation. There's very slight differences here. So Mendel, when he was doing his experiment, found that for the traits he studied, the inheritance of one trait did not influence the inheritance of any other trait. So this law, or the principle of independent assortment, states that the alleles of different genes separate independently of one another during gamete formation. Now this means that a gene that's coded for, talking about Mendel's peas, a gene coded for purple flowers does not always go together with green pea pods. They are going to separate independently of each other. So purple flowers could sometimes have yellow pea pods or green. It doesn't matter, they are independent of each other. So once again, this happens during gamete formation, but it's very important you understand the difference between segregation and independent assortment. Because independent assortment happens based off of two different genes, two different alleles. So in this case, we have what is big S, let's just call that, silver, and we'll call the B here brown eyes. So silver hair, brown eyes. In this case, we're going to take the S's. So the big S here is going to be segregated into different gametes. This S is going to go over here, and this S is going to go over here. Now also don't forget that the B has to go as well. Now these two are independent of each other. The big S and the big B do not have to always go together. So that big B is going to go here, but it's also going to go here. And the little b is going to go here. And that little b will go here. So I know this looks messy at the moment, but do understand that these guys separate independently from each other. They are not correlated, and that is the principle of segregation. That is the principle of independent assortment.